Welcome back. We are going to be talking about your hypothesis. Just as a reminder, if you have not yet completed this slide for background information, stop the video now. Stop this video right here, right now. Fill out your background information, please. Take this time. Do it now. Thank you. Okay, moving on to the hypothesis. So we have this idea. We're going to be making kombucha. We're going to be learning about cell respiration from it. So the question we're really asking is, like, what can we learn about cell respiration from brewing kombucha? So your hypothesis for this lab is that if we brew kombucha, then we will be able to observe anaerobic cellular respiration and aerobic cellular respiration as well. So we have our if-then statement. It's not really like a hypothesis in this lab. We're not figuring anything out. It's more of like a demonstration activity. But that would be our hypothesis. And so then this diagram here is letting you know these different types of fermentation. So different organisms will go through the fermentation process and they will produce slightly different products at the end of their fermentation process. So if you're really into soy sauce, it's fermented by this type of bacteria right here, which is aspergillus. Yes, aspergillus. And so it creates a acid called lactic acid in the fermentation process that gives us soy sauce. In order to make cheese and yogurt, we use a bacteria called lactobacillus, which lacto is talking about milk. That's why lactose intolerant. They're feeding off the sugar in milk, lactose. We have the enzyme lactase. If your body is missing lactase, that means you are lactose intolerant. Okay, anyway, we use lactobacillus to make cheese and yogurt through that fermentation process. And then this bacteria right here, Saccharomyces. Yes, we're going to go with that. It can make a couple different products for us, such as beer, wine, and bread. So for wine, obviously, you need the ethanol alcohol, which is the drinkable alcohol. Of course, you are underage and you should not be consuming these beverages. But part of the beer brewing process, you're also getting some carbon dioxide. That's why it is fizzy. And then for bread, we're not actually containing any of the alcohol that's being produced. We're just capturing the carbon dioxide, which is what makes the bread rise and create like the bubbles inside of bread. So that's just a little background there. Like I said, our hypothesis, if we brew kombucha, then we will be able to observe anaerobic and aerobic cellular respiration. So let's look at the materials section. Again, I'm going to be showing you all of this during the procedure, but we're going to need 1 gallon glass jar, so a really big jar. That's this item right here. You're going to need a 6x6 piece of cloth, like a little cheesecloth. I'll show you. You're going to need a rubber band to hold the cheesecloth on the container. You're going to need, per a gallon, 8 black tea bags. So we need specifically black tea. We're going to need 1 cup of sugar. We're going to need water, both hot and cold water. And then we're going to need 5 to 7 glass bottles and some juice. So, and we're going to need scobies, of course. So that's what we're going to be using. Like I said, when we get to the procedure, I will actually be doing the kombucha. I will be making the kombucha in front of your face, but that's the stuff we are going to be using. So I'm going to pause this video here and I will check back in with you for the actual procedure.